So next up, we have Marko Freese speaking to us. I think, hey, Marko.
Hi, how are you?
Are you in Germany right now?
I am in Bremerhaven, which is in northern Germany, close to the Atlantic, more or less. So
yeah.
We will stay there during my talk.
You what?
We will stay in the Atlantic during my talk.
Okay, all right. Yeah. Well, I'm so glad you could join.
Thank you for having me.
Yeah, great to see you. So Marko is a postdoc researcher in the working group on biodiversity and migratory fishes at Thünen Institute of Fisheries Ecology, and has been working in the Sargasso, mainly looking at European eels. But there's a lot going on in the Sargasso Sea.
That's right.
And not just the eels, and the sunfish have been known to occur there for many years. So I'm so looking forward to hearing what you have to say, and I'll hand it over to you.
Thank you. So I will try to share my presentation now. Let me see how this works. There you go. So could you please confirm if you see it or not?
Looks good, Marko.
Okay, let me see. So that works, right? So yeah, thank you for having me, and thank you for some fantastic and exciting presentations so far. My name is Marko, as what has been said. I'm a postdoc in Bremerhaven, Germany at the Thünen Institute of Fisheries Ecology, and I usually focus on biology and management of diadromous fishes, mostly eel, but also a little bit of salmon. It's my pleasure to present to you the data of sunfish larval distribution in the Sargasso Sea of 2 German-led cruises in 2014 and 15. I will start my presentation with an introduction and by providing you some background information on what we actually are dealing with here. I will tell you a little bit about our study area, what we actually did there, and what we found out about sunfishes in the area. So the Sargasso Sea region is in the Atlantic, and a region that is defined not by the borders of land, but defined by 4 currents, which is the Gulf Stream, the North Atlantic Current, Canary Current, the North Equatorial Current. And the name is derived from sargassum, a brown algae, which can be found in big mats over there. So this is very specific for this region especially, but you'll find that kind of algae almost over the entire globe. The Sargasso Sea is an oligotrophic region, which is characterized by local eddies, and it does have some local upwelling, which can bring some degree of nutrient fluxes in that area. But still, the area, which is surprising, is known for the presence of larval stages of various organisms, including fish, mollusks, and crustaceans such as lobster larvae. And as was indicated before, it's also the famous spawning ground of both Atlantic eel species. And this is more or less also the reason why we got there. The famous Danish researcher Johannes Schmidt discovered the spawning grounds of the eel, the American and the European eel, a long time ago, more than a century ago to be precise, and he came up with the proof that very small or young larvae of the European eel, which were other than in the ocean, are found in the streams and lakes of the coastal areas and inland waters, and that the very small and young larvae of these species can only be found here in this area, in the Sargasso Sea. So this was also the reason for us and my institute, under the lead of my boss, our institute's director, Professor Reinhard Hahnel, to start our studies in this area. And the reason for this is the recruitment numbers of both Atlantic eel species have declined drastically in the past 30 to 40 years, and only little knowledge about the larval abundance in the assumed spawning grounds exists. So there was a lot to think, a lot of things we wanted to find out there. But this was, as Tiani already indicated, not the only thing what Johannes Schmidt discovered. Almost exactly a century ago, actually, in March 1921, Schmidt published a paper in Nature in which he reported the presence of plenty of larvae of sunfish species, which were, as he already knew, I think they were named differently back then, but both species he found were the sunfish, Ranzania laevis, the slender sunfish, and the sharp-tailed molar, Masturus lanceolatus. And he concluded in another paper in 1923 that the Sargasso Sea must be a spawning ground to both of these species. Today, I will here present you the results of the investigations on sunfish larvae that we did for 2 of our now 4 research cruises in the area that our institute has made since 2011. So these 2 cruises are shown here. As you can see, the grids of these 2 cruises are not entirely congruent, as our target area is always the convergence zone of warmer and colder water masses that you can find. So the color of the ocean on the 2 pictures on the right side are indicating the temperature. And well, as you can see, we have a grid of different stations where we sampled. I will come to this in a minute. Anyways, they're not entirely congruent, the areas that we studied, but a large part is more or less overlapping. One difference is that in 2014, with our own research vessel, the Walter Herwig, we steamed back to the Azores and then took another couple of sampling stations there, which we didn't do with the Merian. We boarded the Merian and then left board of the Merian from and to Bermuda. So there we didn't have the chance to go further east. Anyways, this is how we did it. We sampled to collect leptocephali larvae, which were the main focus of the cruise, and other fish larvae. We used an IKMT, an Isaac's Kit midwater trawl, which is a relatively small net with an opening of 6.2 square meters and a length of 10 meters. And we use it with a net mesh size of 0.5 millimeters to also get the small creatures in there. The whole net was towed for 2 to 3 hours behind the boat at a speed of 2.3 knots, more or less. We always had a flow meter in the net to be able to actually calculate the filtered water that we had, because the filter volume was measured by the flow meter, because from station to station, due to weather and surface currents, there's always a little bit of variation. But what we did is we used double oblique holds, which means that we put the net to the water and then we dragged it down to 300 meters at consistent speed, and then we dragged it up again to the surface, back down into the surface. This gives us a good overview of the organisms that can be found between the small organisms that cannot escape the net, but the organisms that can be found between 0 and 300 meters of depth. But that also has a disadvantage, which I will explain later. So what we also did is we collected CTD data to have some abiotic or oceanographic information about the temperature, the salinity, and also the chlorophyll concentration in the area we were actually sampling. So the CTD is more or less a probe that has a lot of different sensors and can tell you, as I just said, salinity, temperature, and all those things at a certain depth. So we let it down to 500 or 1,000 meters, and we can say exactly how the conditions are at a certain depth. So, as I said, originally we were mainly interested in leptocephalus larvae, but I was also interested in the molid larvae. So when the samples came on board from the AKMT, we sorted out the leptocephalus larvae of all anguilliform species, and I also collected a couple of molid larvae from the mixed plankton samples, and then the rest of the mixed plankton samples were fixed in ethanol. Which is not always a good idea, because shrinkage of fish larvae can occur. And since I already collected a fair amount of molid larvae from the samples and measured and photographed them on board, we used those data later to come up with a correction factor, and it turned out that Molid larvae do not shrink as much as other fish, but we published a correction factor for this to be able to actually compare our fixed larvae with freshly caught larvae and measured individuals. So this is what the fixed mixed plankton samples looked like. It was just like a bucket full of living or formerly living things. Which brings us to the research questions, because the data that I will present today have been collected in line with 2 bachelor's theses that I supervised together with some colleagues of mine. Big shout out now to the 2 bachelors, which is Alexander Knorn and Lea Hellenbrecht, who did a fantastic work as they had to sort out all the samples, but I will go into detail to this soon. Anyways, for both of the studies, we applied more or less the same research questions. So we wanted to find out which species of sunfish larvae can be found in the Sargasso Sea. We wanted to know their developmental stages and sizes, and if this can give us an insight to hatching and spawning places and time, and if there are any visible or obvious patterns regarding their abundance in the area when it comes to distribution, and can we find any correlation to the factors, the abiotic factors that we collect with the CTD. And this is where our students came to play. They carefully sorted out every single sunfish larvae out of every station's mixed plankton sample and distinguished them based on their morphology. They identified them, and then, which is fairly easy for Masturus and Ranzania, which are the 2 species that we found there. It's for bigger larvae, it's fairly easy because they can be distinguished by the transversal ribs at the spine base that they have. On top, you can see the transversal ribs, which, as I said, which are absent right here, the real transversal ribs, and they're present in the Masturus. Sorry for the confusion there. Anyways, also the shape is a little bit different. Ranzania is laterally flattened, while Masturus larvae are more or less oval. And when we had very, very small larvae where you couldn't see the transversal ribs, or if they were damaged from the catch, we also genetically tested some of the individuals, more than 150, against morphologically verified reference individuals with DNA barcoding. And just a side note, our morphological based tendencies in both studies were 100% correct. So our students did a quite good job by saying or naming the species of the individuals that we found. So this is what we caught. During both cruises, we found far more Ranzania than Masturus larvae, as you can see here. In 2014, in the Walter Herwig cruise, we found 973 individual Ranzania larvae and 26 Masturus larvae, while we found less of that in the Merian cruise, 365 Ranzanias and 18 Masturus. We cannot really say why the difference between the 2 cruises was so high, that we found almost 3 times as many Ranzania during the Walter Herwig cruise. But one big factor, obviously, were the massive amounts of very small Ranzania that we found in this cruise at some stations. As you can see, the abundance per haul was up to 293 individuals in a single haul, which was not the case in 2015. Also, the size range was different, and Masturus was generally larger, starting at 2-point-something millimeters and going up to 15 millimeters in Molacanto stage. Thank you, Marianne, for introducing that. And for Ranzania, they're really, really small. If you look into the literature, we can assume that some of the larvae we found were more or less freshly hatched. Anyways, we didn't find any clear differences between day and night hauls. However, there was a tendency to catch smaller Ranzania laevis during day and larger larvae during night. This may be a result of different behavior. Maybe the bigger ones are conducting diel vertical migration, that they change their depth during day and night, which probably is not the case for the very small ones, since it's unlikely that they travel a couple of 100 meters when they're only less than a millimeter big. And it's striking that how few Masturus we actually have found compared to Ranzania. Well, this and the fact that we did not find any very small or freshly hatched Masturus and in fact, most of them were already in the Molacanthus stage. That probably means that for Masturus, in contrast to Ranzania, we did not match the exact spawning time or spawning place in both of these cruises. So we can probably go on here. Here's a picture of sheer abundance. If we look at the plain abundance of Masturus in the research cruise area on the right, we can see that they're evenly scattered more or less in the whole research area in this cruise, while Ranzania, by the way, the little dots, the size indicates the abundance and the color indicates the temperature. The more it tends to blue, it's colder, and the more it tends to red, it's warmer. But as I said, you don't find any real connection here to from Masturus abundance to temperature, while there is an indication that Ranzania tend to be in more warmer areas in the southeast. If we look at both of the cruises, disregarding the temperature, we can see in both of the cruises we had really a scattering of the individual larvae of Masturus, which is also a reason why I believe that we did not match the spawning time or place, since most of the time we only had between 1 or 2 or 4 individual larvae in one hole. So yeah, we'll focus a little bit more on Ranzania here. This is a bit different for Ranzania, because here's another view again for both cruises and for abundances of Ranzania larvae in relation to sea surface temperature. We heard something about this earlier as well. Thank you for that. In both cruises, we did find larvae only in or south of the frontal zone, where the warm and colder water masses are connected, which indicates that the warm water masses are probably favored by the larvae and probably also for the adults for spawning. It has to be said though that when it comes to the use of our recorded environmental data, such as temperature, salinity, fluorescence, one issue is that with our sampling strategy between 0 and 300 meters, we cannot really say anything about the depth distribution of our sampled organisms, which makes a correlation with these potential influential factors difficult. Yeah, to answer this in future, stratified catches would be great. If you look at the salinity, though, you can see that the tendency for Ranzania was there to favor more saline water, which was also found in both cruises. The study with the most and smallest collected Ranzania, Walter Herbig 373, as that you see here, and we found very high abundances with relative abundance of more than 10 individuals only at stations with sea surface salinity exceeding 37 practical salinity units. This doesn't give a full picture, but at least give us an idea that this may be a factor. So I'm coming to an end now. I see I'm 1 minute 40 over. Anyways, these are the conclusions and outlook. So the question was, which species do we find? We did find Ranzania laevis and Masturus lanceolatus larvae. We did find comparably large numbers of Ranzania and rather few Masturus larvae between mid-March and end of April. And for the question, what are their developmental stages and can this give us insight to hatching and spawning time? I believe we have found spawning or recent spawning of Ranzania in the area, because throughout the cruise we found very small freshly hatched Ranzania larvae, which does confirm the Sargasso Sea as a spawning area for this species. And then when it comes to the visible patterns, apparently Ranzania uses warmer water, denser water, south of the convergence zone. But Masturus seems to spawn at slightly different place and time, since they were scattered all over. For the outlook, I can say it would be nice in future cruises if we can conduct stratified sampling studies to have a better view on the correlations with abiotic factors. And it would also be nice to have sampling directly in the Gulf Stream to understand more about the recruitment mechanisms behind this, because the Sargasso Sea is centered in the middle of these currents, and it would be interesting how they use the currents. Thank you. So a big kiss to those who contributed, especially my boss, Reinhold Hanel, Lea, Alexander, my colleague Jan Dark, Tina, Lasse, Klaus, and Hakan Wikström, and of course the crews of both cruises. Thank you.
Oh, thank you so much, Mark. we're going to have John Houghton come back on. Just so fascinating, and to be able to get the larvae so fresh at that extended period of time with the metadata. I mean, that's the big thing, is the metadata that goes along with the larval collections. So important for trying to figure out who's doing what. Yeah.
Yeah. Sorry for being a little bit too long in my presentation, but I guess that was fine. So. Yeah.
No worries. When's your next? You're going out. Well, I mean, COVID willing, but I know you had one in March. And any prospects as when that might go out? Your next.
Yeah. We actually had a cruise planned in March last year, which was canceled entirely due to the COVID pandemic. It was just a matter of days. My boss was almost always already flying to the Bermuda, and then on short notice they canceled the trip, and the entire cruise was canceled because we couldn't go for several reasons. So anyways, the next official cruise in line with our monitoring of the eel larvae would be in 2023, I think. Yes, 2023. And there might be another cruise earlier than that will not focus in the area of the Sargasso Sea, but on the continental slopes of North Africa and Europe, which we will also use to look for larvae, even though our assumption is that we won't find any molid larvae. But this would also help us to understand how the whole current system works there and why those larvae are not distributed in that area.
Right. And where are the mola genus?
Exactly, yeah.
It's so fascinating. The Sargasso is such an interesting area because it's a mobile address. You know, it's moving and it's changing significantly with climate change as well.
And it's large. And it's really, really large.
In some areas causing real problems in the Yucatan, for instance, with the huge amounts of sargassum washing up on the beaches there. Yeah, really, really interesting what's going on in that whole region. Well, John, did you have any questions?
Super quick question. Thank you for doing this because I've been wondering about it for years. And that's lovely talk, man. It doesn't matter if you went over, that was worth every second. That was great. What's the potential for will these kind of data be integrated into long-term sampling protocols for your institute, or is this a short-term project? Do you think it can run long-term? Because it's huge effort.
Well, to be honest, I would like to do it because, as I said, the most important thing that we're doing there is finding out more about the abundance of eel larvae in the area, because this is the whole management of these species is based on recruitment data of glass eels at the continental slopes of Europe, and we don't have any idea about what is going on in the actual spawning area. So we will continue doing this and trying to deepen our understanding in the biology, the reproductive biology of the eels. But also we have these samples, and I always said, well, as well as my whole team and my boss, that it would be interesting to also get as much information as possible from these cruises. And since already Johannes Schmidt found out that those molid species were probably spawning there, I found it almost as exciting, and then I found people that helped me with it. So what Jenny just said with mola, for example, what I find interesting is there were like a few records of mola larvae in the Gulf Stream north of the Sargasso Sea, but we didn't find any in the Sargasso Sea, which for me again is just a mystery, because how do they actually do it? Other organisms at the continental slope, such as lobsters, they release their eggs into the current and they get transported into the Sargasso Sea without moving by themselves. So it's very interesting to see how these mechanisms work in the area.
Yeah, and we have substantial populations of sunfish on both sides of the Atlantic. So doing latitudinal migrations, and I mean they're releasing their eggs somewhere.
And I didn't really properly answer the question. I think we will continue doing this, and I will do my very best to find the time and people that help me put the effort into analyze the data also from the other cruises, because we have still one cruise that we didn't analyze yet, and we didn't collect and count the Molid larvae from. And I hope we will find the time to do this and continue to do this.
Well,
Yeah.
if you ever need as a support to funders or anybody, I'd write one for you in a heartbeat, because I think that's fascinating. But just the last comment, it's just so cool living on the island of Ireland, because it's always been, we've got Loch Ness up in Northern Ireland, which is one of the largest eel, grounds in Europe. So it's always fascinating me, that connectivity between Northern Ireland and the Sargasso, and then you suddenly go, Sunfish can't be doing something weird as well. It's just, yeah, no, I love that, man. That was really cool.
Yeah, I love it as well. I actually have been to Loch Ness before for a meeting.
On a European scale, that's a big lake.
Yeah.
Well, it is for us anyway, not for anyone else probably in the world, but yeah, it is for us. But no, it's a cool place.
It is. I enjoyed it very much.
Very good, very good. Superb. Right.
More mysteries to solve. Well, thank you so much, Marko. I hope you stay tuned in for the rest of the symposium.
Of course. Yeah. Thanks for having me.